Okay, welcome back again.
So if you don't mind, Rafael, I'm going to just briefly introduce you. So Professor Rafael Bernardi from the Department of Physics at Auburn University. He was a so long associate of our center. Actually, he's still associated with our center with extensive experience in polling simulations, working very closely with experiments where they do similar treatment of the protein, interface of QMMM in NAMD, and also development of tools in VMD and NAMD that allow you to better use the software and set up simulations analyzing with QwikMD as you see in his title slide and so on. So he has extensive experience applying our MD simulation and VMD and NAMD specifically in very close connection to experiments. So he kindly is involved in our training activities. Thank you very much, Rafael. It's a pleasure to have you here. Please.
Thanks a lot. I missed the first part of what you were saying because my computer was having a problem. I got you're talking about me, so like, thank you.
Yeah, of course. Go ahead.
No, I'm just joking. Like I heard the second half. So I don't know if the first half is true, but the second half I will try to show that I have some.
No, I'm talking about, you know, that was.
So yeah, what I want to show to you today is a little bit about QwikMD and how we developed a tool to make your life doing the simulations a little bit easier. And the whole idea behind it was we were initially like a few years ago we were concerned like why would some user prefer one software over the other? Why would a user prefer, like, what are the troubles that the user might have in the beginning? Like, there are questions like that, you know, like things that what motivates people to use MD, like when they're experimentalists. And we were trying to develop then a tool that would work somewhat like a kit that you have in the experimental lab that you can apply very easily to your problem and solve most of your regular problems that you have doing simulations, while trying not to make it a black box. You know, like that's two things that we have in mind. So I'm going to show you a little bit about it today, and you're going to have a chance to play with it this afternoon if you haven't had before. So yeah, like, so QwikMD then is just like I was just summarizing that, like this is a tool that helps you to do that. It's inside VMD, how to do simulations. It's inside VMD. It connects you directly to NAMD so that you can do the simulations. You can even do it live. It provides some info buttons, and these info buttons have even links for papers and things like that. So it helps you to understand what you're doing, why you're doing that way, and give you some references about like where to look for more information about what you want to do. And the whole idea behind it was like to make it as complete as possible. Of course, it's not there yet, but like it's very much useful for most of the things that we do on a daily basis. But the idea was basically that you could use it for everything that is more that does not involve any development, and you could use the software like for most of the MD simulations. The reason why we care about that, I always like to put this sentence, you know, like it's because we really care about understanding biology for understanding how the atoms jiggle and winkles, you know, like we want to understand how that happens, and we want to create tools that make that easy to do. And over the years, there was a lot of evolution in that, you know, like for people to start studying this kind of like studying proteins with you learn about that before, we learned about that in the last couple of days. They started to study this before computers became a thing, you know, like, and they were using, like, for instance, here you have Lord Kendrew, like with balls and sticks trying to get the whole structure of hemoglobin. And it was much different than what we in the 90s when we started to use computers more frequently for that. And we got to points like in recent years where without computer you cannot think about solving these structures because they get too complex. So how can you then use a simple tool? How can you develop a simple tool to help with all these steps? You want to get to this level here, like to the 2013 level here that I show. So QwikMD basically follows this workflow where you start from the protein data bank. If you have a homology structure, you can also start from that. You know, there's no problem. If you have your own structure that you just solved in your experimental lab, great, you can start from there. But like in principle, let's pretend that we are starting, let's assume here that you're starting from the protein data bank. So what you need to know the PDB code of your structure. And if you find that code there, like in the protein data bank, you can get, for instance, this structure here, which is in this case is strep-avidin. And you can have the strep-avidin here is a tetramer. You can come and select what chains and molecules you care about in your simulation. For instance, I want you to study the interaction between biotin here and strep-avidin, and I want you to simulate both. And or no, I just want to simulate one of them. Or no, I don't want to simulate the tetramer. I want to simulate a dimer, or I want to simulate just a monomer. You can use that. You can use QwikMD to select the parts that you want to simulate very easily. I'm going to show. Then you check the structure. And when I say check the structure, is there are many things that goes here. Like there's a lot of automated checks. For instance, we check if there are force field parameters available, if there is any like outliers in the Hamilton plot, you know, like of the protein. We check for gaps in the protein sequence. We check for quite a few things, and like that are more common to happen when you're opening a structure for the first time. There are a few things that we want to add that are still not there. For instance, checking protonation states and things like that. But like that's hopefully we're going to be able to add that at some point. Then you can have a step of visualization. Like QwikMD is going to help you to load the structure in a more beautiful way. Like the way of doing that is instead of using lines and sticks, like typically the VMD load structure, it already shows the secondary structure for the proteins, already going to show ligands as van der Waals. It's going to already show water molecules in the way that you can understand what they are. It's going to do all of that automatically, select things the way you want. And that is done also because QwikMD is not, it was designed not to handle 100 million atom systems, you know, like, so you can do that. You know, that's the reason why the VMD regularly don't do that, because if you try to load an 100 million atom system already showing balls, like spheres, like van der Waals, or a surface, that's going to crash the software. So we can do that because QwikMD was designed for a smaller system. Then continue, you can select protonation states. Like I say, like sorry, point limitations. That's the first part here. It's like point limitations. For instance, if you have an enzyme and it was crystallized with a modification in the catalytic site, because you know if you don't do that, you don't get the co-crystal because your enzyme is working. And so what you do then, you go there and say like, oh, actually, this is glutamic acid here, was not supposed to be a glutamic acid, in this case probably was supposed to be a glutamic acid, was they crystallized with something else there, like an alanine. You go there and you can mutate. You just click and select the residue that you want to do that. QwikMD is going to do that for you. If you start mutating everything, QwikMD is going to warn you that that's not exactly how things are supposed to do. You know, like, so if you try to change the sequence of the whole protein, it's going to tell you that's not how things happen, and it's going to provide some warnings and explain to you what's happening. Then you can select the protonation states, like I was saying. We would like to have at some point some automated way of calculating that, but as of now, you go there and you select yourself by hand, and that can be very, like, you know, for many reasons, it can be very important. But for instance, here we have a chemical reaction, and one of these enzymes is donating a proton. So if you don't have this histidine here in the protonated state, the whole chain here of reaction is not going to happen. So QwikMD helps you to decide that. But if you look at the structure, you have the sequence, you can go there and click and select the atoms, and you can easily do these mutations. Then you're going to solvate. And for solvating, then you can select, I want a water box, I want a membrane environment, and QwikMD is going to help you to prepare all of that. So for water boxes, it's easy. You're going to say, I want this size, or like not even the size of the box, but like how much space you want between the protein and the box. If you are in the R-Easy run, that's the simple version of it. It's like, I don't really know what to do. It selects for you a standard, a kind of a safe distance that is actually even larger than most of us used to run. just to make sure that you're in the safe spot. If you go to advanced mode, you can then select how much space you want. If you then want to do a membrane, which is only in advanced mode, you can go there, select the lipids. I think you only have 2 right now, lipids. You can go there, select the lipids. It creates a flat membrane. You go there, insert the protein in the pore. You can move things up and down. It's very fast because before you actually, it does not generate the membrane for you right away. It just show you a box, like something like what I have here, like this box outside of the membrane. It's going to be where the membrane is. It is going to help you to guide your protein inside the membrane. And then after you select, like, okay, should be something like this, you click another button and it generates the membrane. Then the other thing you can do is to select the salt concentration. For instance, you want to add ions in that system. So I have here my water box without any ions. This is going to be, I actually want a solution of sodium chloride. You can have that. You want a solution of something else, you can have that. A potassium chloride solution, you can have it. And it helps, like it creates all of that for you. Just say the concentration that you want. Then you can select the protocols. What do you actually want to do in the simulation? You can just do the, let's say, the vanilla MD, or just run this, and can do that for you. That's available also in the easy mode, and also the QwikMD dynamics is also available in the simple mode of QwikMD. If you go to advanced mode, you can do QMMM, you can do molecular dynamics flexible feeding, you can do all the protocols that are more complex, and all of that is simple. Like you just select a few parameters and it goes. Many of them are pre-selected, like in the default value that are reasonable, and you can, in the info button, it explains to you what numbers to select. Then you can also select things like, I want to do this in MVE, I want to do like in a you probably heard about that in the last couple of days. The molecular dynamics ensemble that you want to simulate, you want to simulate an ensemble where the number of atoms, the number of particles, the volume and the energy is constant, or instead of the energy, the temperature is constant, or pressure, and number of atoms, pressure, temperature is constant. So you're going to select what you want. You're going to select the barostat, you're going to select the thermostat, you're going to select things like that. QwikMD is going to give you all the options, kind of standard options, and you can go there and change if you want. And you can make selections for basically every step of the simulation. As soon as you do all of that, you can prepare your files, and it's going to generate all the files exactly the same way you will do everything by hand. You're going to have all your dynamic configuration file, you're going to have everything prepared the way you need, and all of that is going to be all of that is going to be in one standalone folder. What that means is QwikMD is actually going to generate 2 folders. One is called setup, the other one is called run. So the setup folder is where everything is created. All the scripts that were used to generate the files are going to be there. Nothing's hidden from you. And then the run folder is going to have everything that is needed to run. So if you want to run in a supercomputer, you just take this run folder, take it there, you can run it. You can run on another computer, you can take it. Whatever you want to run, you can do that. If you want to prepare everything in Windows and run in Linux, you can do it. You just take this run folder and you're going to be able to run. So everything is going to be there, and everything is going to be saved in 2 log files. So the 2 log files, one of them is what we call a human readable version, and that one is basically telling you, you selected this temperature, you selected this ensemble, you use this method, give you the references. You're using SMD, this is the reference for SMD. You're using MDFF, this is the reference for MDFF. It's like something that you can almost copy and paste to a method section of your paper. Like it's not perfect, the text, but it gives you all the parts. You just put the words that you need to link the things together. Like it makes your life easier. The other log file is a TCL script. It generates everything you did, every selection you did here, like during the process, it's going to be in that TCL script. And if you run that script again, like in VMD, it generates everything you did. So it's going to repeat every step that you made to get to that point. Any questions? No? You just keep the chat open. If you also want to ask something in the chat, feel free to do. So after everything is prepared, and when you click prepare, is that when everything happens, you go to the step where you're going to run. But before that, just let me show you how QwikMD looks like, in case you've never seen it. It's a window in VMD like this. You have the easy run and advanced run. You also have some analysis. I'm going to talk about it later. So as soon as you type here the PDB that you want, you can click load. That's going to load the protein. For instance, here you're loading a protein where you have protein and water. It's going to separate that. It's going to represent the protein, like here in the cartoon. You can change or van der Waals. You can select then, let's see if it's an NMR structure that you have many steps. You can go there and select the step that you want to simulate. You start from. You can select the chain. For instance, I just don't want, like I was showing before, you have a tetramer. I just want one part. You can go there and select the part that you want. You can do some structural manipulation. The structural manipulation is where all the checks that were made are going to show here. And then you can do a mutation, like here. I want to change this leucine to a threonine. You can do that here. And you can select the emission states. You can change the serine here to whatever residue you want. It's like I mentioned, like how the checks are here. Here it's showing the colors that represent the secondary structure of that part. And also, let's say you have a ligand that you don't have parameters for. You can come here, add topology and parameter files. And if you get those parameter files yourself, you can add them. If you use FFTK, you can just add them here. If you didn't do it before, you can come here, select, it can open FFTK for you, and you can generate that yourself. You can also open automated tools that are online to do that. It's going to just call these automated tools and get you some structure. You can also use it to call manufacturer to design your own molecule if you need, like a ligand, if you want to make a change in the molecule that is there. All of that is connected through QwikMD. Okay, so after you did all of that, you can come here and select, I want explicit solvent, like I was saying, you want to solvate the system. I want this salt concentration. That's the salt that I want. I want to run equilibration and MD. We are all in the easy run here. In the advanced run, there are many more options. You select the temperature. It's going to show also in Celsius and Kelvin. You can select the simulation time. And then you're going to select where you want to save it. And as soon as you hit prepare, all those scripts and everything is going to be generated where you selected to save. And in this case here, we are selecting live view. So if you're selecting live view, when you're running, it calls VMD and shows live in VMD, so you can see the atoms moving at the same time. Okay. So after you were done with that, you can go to the next step where you start the simulation. So start the simulation, you can select, I want to run that simulation. In the desktop, I was doing everything. I want to run that simulation in a supercomputer. You can copy the run folder like I mentioned, go to a supercomputer, run it there. Or you can also run it live, like I was showing you, and you can see it, like in your computer, you can see this moving in front of you live, like the simulation that you're calculating and seeing at the same time. That slows down a little bit the calculation, so I would only use that for showing someone, like if you're in class, you want to show someone like how to do those things. Or if you want to test something, like, and you want to check at the same time, it might be useful. But like, don't do that for a production run. That's going to take a month running. Like, that makes no sense. But like anything that you want to test, it's also a good platform to test because you can see it right away. That's it. You don't know why it's crashing because it's complaining that something's happened. You can use this to go there and check what atoms are flying away or something like that. You can use that as a tool to help you understand what's happening. You can do, if you're running live simulations, you can do live analysis. So you can check RMSD, you can check number of hydrogen bonds, you can check distance between parts live, but you can do that while you're doing the simulation. And there are some more advanced analysis that you can do afterwards. And some of them can give you like maps of hydrogen bonds. Like, it does not give you the beautiful picture here that you need some Python skills to get it. But like basically, it gives you all the data that you can make figures like that. It's going to give you all the information that you need. In this analysis, you can see things like here, for instance, I'm looking at the energy. You can track how the energy changes over time. You can select, like, you can do many analyses at the same time. So you want to do RMSD, energy, some thermodynamics like temperature, and you generate different tabs here, and they're calculating all of them at once. So if you are live or if you're not live, it can create all of these tabs. And you can export those then as files that you want to use as an image, or even like an CSV file that you can open somewhere in some other codes. Okay. After, okay. Question? No. Really? Okay. Continue. What can you do with it? So these are the benchmarks when we published QwikMD back in 2016. So that's what you could do. So you're assuming like a lot of these users are actually not users of supercomputer centers. And you were like, okay, let's see what you can do with a desktop or a laptop. You know, so these were numbers that we were getting for these systems using, like for instance, a MacBook Pro from 2013, you know, like or a desktop, something like with 8 cores and a NVIDIA GeForce Titan X, which is like an old NVIDIA right now, and or using the cloud, you can use QwikMD in the Amazon cloud. It's available there, so like in the AWS. So we were testing those numbers, and you could see here, like, you know, like for systems that most people study, like in the 100,000 atom range, you could get like more than 10 nanoseconds a day. You know, like that was kind of a number back in 2016 when we published this. Now you can do much more, and I can show you some of the most recent results. So if you want to use, for instance, one dual Xeon machine with 32 CPUs and a Titan RTX, like which is not even the top machine right now, you can do a 100,000 atom system using the standard 2 femtosecond time step, 12 angles cutoff, the standard Charm force field parameters, not cheating, not trying anything special here to go faster. With NAMD 2.14, you can do 28 nanoseconds per day. And you probably heard a little bit about NAMD 3. With NAMD 3, you can get 87 nanoseconds per day with the system. So you can do a lot, but with not even the newest models of GPUs. If we go to newer models, like if you use an RTX 3090, then here you have for many different sizes. I just have some numbers here for you. If you get, for instance, a machine with 2 3090s and 80,000 atoms, you can do almost 500 nanoseconds per day. And this is now using 8 angstroms cutoff. Like you're using more like an Umber set of parameters. Should I use that all the time? It's up to you. You know, like some of these parameters are normal. You can use it. Like there are many literature showing that you can use, like 4 femtosecond time step is becoming a new standard. But some of these, like the 8 angstrom cutoff, there are some work showing that, you know, like it works for some cases, it does not work for everything. So be careful when you're selecting. This is just to show you how fast you can go. What's the speed of light today in MD, you know, using our software. If you have a more powerful computer, if you have like a DGX A100, you can get even systems with like 7 million atoms, you can get more than 20 nanoseconds a day in one node, which is a pretty impressive number. And all of these simulations can be prepared with QwikMD. Okay, so you can prepare with QwikMD and run this and get this number the same. Like this, actually, for all these benchmarks, I use QwikMD. Questions? No? So, okay. Why do we want all this computer power? What kind of things we can study with it? For instance, you can use all these powerful computers together with QwikMD to study the stability of lipid-protein interaction. Like in this case here, we are looking at the respiratory complex. In the respiratory complex, you have some lipids that are kind of almost integrated to the protein. You can calculate the ones that are always in contact. They're always there. They're always kind of part of this interface between protein and membrane. You can also calculate things like allosteric communication within the protein complex. And you can see here how, for instance, the part where the NADH binds to the respiratory complex interacts with the quinone here at the bottom, going interaction through a chain of iron sulfur complexes. So you can use this tool that we're showing and to study this kind of mechanism. So I myself have used it a lot, like I mentioned that I use it a lot for SMD, like for studying mechanoproteins. And I'm mostly interested in studying how many mechanoprocesses happen. Like I started doing that, you study ion channels, like I was studying a potassium channel that was interact with the membrane. So I was kind of using it for studying how to gate the channel, like how to open and close. That was part of my thesis. But then after that, I started using it more for studying proteins that are the mechanical properties of those proteins. Like for instance, like if you make a mutation, make it weaker, those mutations can make the protein more sensitive to force application, and that sensitivity affects biochemical signaling, and it can create some diseases. For instance, this happens with filamines, and there are some genetic diseases that are associated with that. That in collaboration with Ulla from Finland, also I've been studying many other processes where force, like so some of these proteins, they are very strong. They resist your force, like they can resist your force in a very strong way. Like they can diverge force so that they can withstand very high amount of forces and high amount of force. And then I'm studying how that happens and how they are able to create force pathways that make them stable at high force conditions. And I'm going to quickly show that to you just to show how somebody asked here, but the step is 4 femtoseconds. I think you're talking about this one. Yes, this one, the step is 4 femtoseconds. That was the question. So how do we measure force? And so just to show, Emma mentioned that I work a lot with people doing experiments in this, like in force spectroscopy, and I'm going to show some of the results we have combining the two back then. So I'm getting questions about the 4 femtosecond. So how do you get that? There are multiple ways of doing that. The most used way right now is you use what we call hydrogen mass repetitioning. And basically, what you have is the reason why we use like a 0.5 femtosecond time step, which would be the tradition, the right way of doing it, was because the vibration of hydrogen atoms, like that's the fast vibration you have, like the vibration of the bond containing hydrogen atoms, like it takes about, I don't remember the exact number now, but like in like 0.5 is about like 1/10 of the whole vibration. And so you need to have something that you can break in steps enough so that when you're calculating where the hydrogen atom is going to be in the next step, so they're vibrating, my next step is going to be here. If I go a step that is too long, if I'm having that force, I might go all the way here and I'm out. So I'm creating something that is not realistic. So you have to have a very small step so that when you're vibrating, you're actually discretizing the time in a way that you can simulate what you're vibrating, what you have vibrating. So then we created some methods for restraining it and not having that vibration, but like correcting for that. And then we were able to do 2 femtosecond time step. Now, to do it for, what people have been doing is to redistribute the mass of groups. For instance, you have a CH3 and you have 3 hydrogens in the carbon, you redistributed the mass of the carbon to the hydrogens. And then when you do that, you end up slowing down all these vibrations. And by doing that, you are able to increase the time step. Because at the end, the whole CH3 still have the same mass, the assumption is that that's not going to affect too much. And there's a lot of papers showing that for most properties, that's okay. You can do that, like if you want to study most things that you want to study with MD. So it has become a new normal. Can you do that with QwikMD? It's not there yet. So QwikMD is not there yet to do that, but it is already in the PSF gen. So if you generate everything with QwikMD, you only need one command from VMD to convert that to a 4 femtosecond time step. So there is a command in the PSF gen that you can convert any PSF that was generated for a 2 femtosecond time step to a 4 femtosecond time step. That's a very easy thing to do. It's not already inside QwikMD, something that we need to add. Any other question? Okay, so that's what I do. No problem. Okay, so I'm interested, like I say, in measuring force and measuring force in this protein. So I work a lot with experimentalists. So how do they actually measure what I'm going to measure? So they can use, I work with people using magnetic tweezers, optical tweezers, and atomic force microscope, mostly with atomic force microscope. But what they do at the end is they have a protein of interest that they want to study. They have a lot of chemistry magic here to make sure that the protein is tagged to a surface, and then it's tagged to, like in the magnetics tweezer case, they have it tagged to a magnetic bead, so that they have a magnetic field, so they can pull it, so they can move this bead outside the magnetic field, so they can track it. Okay, they can track how this bead is moving. They know the force based on the size of the field that they have, so they can measure how the system is reacting to that force. The same way you can do optical tweezers, you can trap these beads with optical lasers, so like you can trap them, and then you can pull the system, like the beads are connected to the protein, and you can pull the protein, and you see how the protein reacts to force. Same thing with the FM, you tag it, like you tag it to a surface, you put it at the glass surface, and then you have another tip of this protein interacting with the cantilever of the FM. So when you pull it up and down, that's gonna change, like you're gonna reflect kind of like the position of the lever. So there's a laser hitting that, and then you're gonna measure how the laser moves in a detector, so you know how much change the shape of the lever, and because you know the constant of lever as a spring, you can calculate the force. So that's how that's done experimentally in simple words. You can also use something new that I actually don't collaborate with anyone doing it, and that's, I think there's only one lab actually doing it, which is a very nice technique. It's called centrifugal force microscope, and what they have is they spin a microscope very fast. They have many beads at the same time, so they do single molecule experiments, but they do like thousands of single molecule experiments at once, because they have many beads. They can track each one of them individually, so it's single molecule, but they do all at once, so they get ensemble data out of, not an ensemble data, they got multiple data out of, they get a large N out of one shot only, like instead of doing it multiple times like they have to do in the other ones. And by doing that, they can track, like by knowing the centrifugal force, they know how much the bead is going away, holding protein, so they can recover the force that the protein is actually feeling. So, okay, so what I do then is, if the AFM can pull the protein, let's say you want to pull interface molecule 1, molecule 2 here, and the simulations, we can do the same. We can hold one end, pull the other. Emad was talking about that earlier. So you can hold one end of the protein, pull the other end. In my case, I'm not interested in recovering free energy because I'm interested in the system at high forces. So then I don't go slow. I go very fast, actually. I go at high force level. And so what you get at the end, we get force extension curve. Experiment gets exactly the same thing. We can compare the two. They agree. Typically, that's what happens. But then the experiment only gets that, the experiment, if any other thing, if they want to test anything, they need to go back to the bench, mutate the protein, and get a different trace for a mutation, because they have this hypothesis that amino acid is important for some reason. So they can go back and do the experiment again. In the simulation, we can see what every atom is all the time, and then we can understand better the system, and we can design the new experiments. We can design even we can actually create a theory why that's happening, like why your system is strong or weak or whatever is happening with the system. And we can use that to guide new experiments that can prove what we did, and can also guide them through a more like intelligent design of stronger systems or weaker systems, whatever you're interested in. Okay. So what we have been doing then is, at the end, we have the same force field that if you run the simulation that you've seen before. We have a term for bond that's going to vibrate. We have a term for angles vibrating. We have the same terms that you learned many times, probably in this school so far. See, you're going to have angles vibrating, like the second term here. But then we also add another term. We add a term that is basically the hook potential, which is just a spring that we're pulling the system. So we're holding one end, have this hook potential pulling in the other way. So because we know the K, we know the constant of the spring, and we know we are going to measure all the time how much delta X is, we know the force. Okay, so we're going to know the force that you're pulling the system. Of course, it looks very simple when I just add one simple term at the end of this more complex equation, but you have to remember that we are dealing with like thousands of atoms, like in the most of my case, like in the simple small system, we are dealing with 300,000 atoms. A lot of the times we are dealing with 2 to 3 million atoms. So you have to remember that all these things are moving at the same time. So we use NAMD, VMD, QwikMD all together. So these numbers here are wrong, they're old. Probably heard about some new numbers. But like we're using NAMD, QwikMD, and VMD to do all of that. And here are some of the results we have. So this is one of the systems we study. It's a multi-million atom system called cellulosome. We're particularly interested in the cohesion Dockering interface. We're particularly interested in how this large system are formed. So all these systems are basically formed by these interactions that are not non-covalent. And as you can see, this is very large. It's sitting outside of a bacterial membrane. And so this can withstand a lot. Like this is actually, it can receive a lot of force because this molecule interacts with the food of the bacteria. So the bacteria wants to use, this bacteria particularly wants to use plant fiber as their food source. And so it attaches to the plant fiber, starts degrading it. And at the same time, you are in the kind of a crazy environment where there's a lot of flow. And basically you are holding into the food by these atoms, so by these bonds that are non-covalent. So if you're doing that, you can imagine that these things need to be strong. And so you're interested in studying them. And some other systems that we study, like in the same complex, we study how each one of the parts of that scaffold that you have there are interacting. And we're also studying that using the same methods. And the interesting thing about this specific system is we're studying how each one of them, how strong each one of them were. And by doing an analysis like in the basically, like we had to make a model. There was no model. We had to create a model using modeler. We created the structure for each one of the system. We studied each one of them. There were 7 there, so there are 7 domains here. So each one of those domains, and we were able to see that different domains would resist to force differently, and that would depend on the position where they are. You know, like the ones that are closer here, they can resist to more force, and when they're here, it is less force. And when we start to use the techniques that we have, you know, like we could simulate the pooling, so we're simulating the unfolding of that system. We could unfold the system with the simulations here, the experiment here, because we're pulling much faster because of the time. But like we recover the same like Bell-Evans curve here, and what we get at the end is that we can explain what's happening in this experiment to the point that I could understand why they are different, and also suggest mutations to the weaker one so that it would become stronger. And that's the most interesting part. So yeah, here's just a correlation between experiment and simulation. For one of them, our model was not good. For the other 6 was good. And the simulations were able to tell this region here is what is making this cohesion number 1 weaker. So if we come to that cohesion number 1 and mutate that area, we're going to find a stronger protein. So what we did then is to see how we could mutate that in the computer. And we found that, okay, doing this mutation here, you can see that what is stronger, what is weaker. And we suggest that to the experiment. And, okay, it's not showing here. So, and at the end, the experiment showed that the mutation that we suggested, this one here, A105G, actually this is the simulation versus the experiment. The experiment showed that this was 2.6-fold stronger. So we could make a system 2.6-fold stronger based on the calculations we did from the analysis of this FMD. All of this, like I say, back to QwikMD, all of this created with QwikMD. So all these systems were prepared there. There's nothing special about what we did other than some more maybe complex analysis, but like all the simulations were done inside QwikMD. So yeah, here there's some other systems, some other cases. I just gonna go fast through them because I wanna go to another part of my talk. But like we were studying also more like stronger system. Particularly, we were interested in studying bacterial infection, how that can be studied with the computer. So we were interested in studying how the bacteria interact with the human cells. So here you have a protein from the bacteria, and here is a protein from our cells. And basically, when you have staph infection, that's the initial step. So the first step is when the staph goes inside you because of a surgery or something. The staph is going to interact with your extracellular matrix by these adenosine proteins. And so we want to study that process that's supposed to be relatively strong. And we end up finding that this is the strongest protein interaction known, and by far, and to the point that although this has no covalent bond, this was as strong as a covalent bond. And so we did thousands of simulations. So these are all results from simulations. So as you see, each one of these points here is a different simulation. There are 3,000 of them. This is what we recover from the simulation. So the distribution of the forces at different velocities. So like Emad was showing, you do SMD, you can pull in different speeds. So this is pulling at different speeds, and then we can recover the data from the experiment. And I want to just point out that these are excluded from the feed. So these are my experimental data excluded from the feed. So I feed only my simulation data, and I perfectly recover the data from the experiment. So when we do that, we know for sure that what we are doing is what we are measuring in the experiment. And because of that, we are able to explain exactly why that system is strong. So we can test hypotheses. We can test that, for instance, these bulky amino acids, are them the reason why? We show that they're not, like they believe to be the reason why this is strong. And we are able to show that a network of hydrogen bonds is actually what makes the system strong, which is not that impressive if you think about it. But there is one reason why they're impressive. It's because they form this corkscrew shape. So like they go kind of rotating. And because of that shape, doesn't matter how you pull it, you always end up aligning the system. Because like, remember, like if you have just bonds aligned like this, and this is an IG fold that happens all the time, if you pull like this, you're breaking one hydrogen bond at a time. If they're twisted, doesn't matter how you pull, you always end up aligning. You always end up having a lot of hydrogen bonds aligned. When you align the system again, you're always in the perfect shear geometry. And so the system needs to break all the bonds at once, otherwise you don't break it. And because you're breaking like 10, 15 hydrogen bonds at once, you end up having the strength of a covalent bond. So you can have a covalent bond interaction level, you know, like in terms of force, without having the cost of producing this covalent bond, like without having a sortase that goes there and make the two glue together forever. You know, so it's a very interesting mechanism. The more interesting part is it only uses hydrogen. It only uses hydrogen bonds between backbones. You know, it's a beta sheet. That's kind of what they do. But the thing that is nice about the computers is we can go in the computers, kill all the other interactions, and just make it as a glycine chain. You know, they can show that they're still very strong, even though we have absolutely no side chain. And say, okay, that's crazy. You cannot do that experimentally, but you were lucky enough to have a homolog system that was only glycine and serine, so also almost no side chains, and it was as strong as the system that we. And then we look at all the possible like homologs, and we saw that all of them were in the same level. So they are all in this 2 nanonewton regime of strength, which is like the regime of a covalent bond. So this is by far the strongest protein interaction known to date. You know, like to compare, a lot of people think about strep-avidin biotin as the strongest interaction, and strep-avidin biotin, which was considered the strongest until like 5-6 years ago, is like less than a tenth of this. So like it's much weaker, like an order of magnitude stronger than one. Okay, so using all that information, you can design the ligands, and again, all of this done with QwikMD. I can prepare a system like that in QwikMD in 1 minute in front of you. There's nothing special about it. It's done here, other than thinking about problems, how to plan the experiment. But you can do all of that using QwikMD, and that's what my main message for that. But what else you can do with QwikMD? If you're not interested in pulling things like I am, you can use QM. You can study chemical reactions, like you can produce orbital images like this one. You can study chemical reactions like the one I mentioned before. You can study like this whole process here, how all this chemical reaction happens. That can be made with QwikMD very easily. You can select, for instance, if you want to do a QM calculation, like I have that image before, but without the QM, you can now come here after classical simulation and select what's the QM region, what is the quantum mechanics region that you want to simulate, what is the protocol you want to use, what are the methods, the exactly level of the simulation that you want to study that part. You can, do you want to save the orbitals for looking at them later? Do you want to track how the orbitals move? You can select all of that with QwikMD. And then you can also study, like for instance, here you have the orbitals of one molecule. You can study how their energy evolve over time. So here you have the highest occupied molecular orbital. You can see how they evolve. You can see the other orbitals as well, the non-occupied orbitals. You can generate nice image like that or rotating too fast, but like this is just a DNA helix with this 3 base pairs simulated in QM. All the rest is classical. Combining with what Emad was showing, you can study very complex processes. Like for instance, you want to study how the tRNA synthetase works, like one of the most important enzymes for like setting our genetic code. You can see how the tRNA binds, that all of that can be done with classical simulation, but then you want to study how you're charging the tRNA, but how you're adding, like in this case, a glutamate to the tRNA. And you can look at that, and we look exactly into that system. You can look at that using QMMM, using QwikMD to prepare it. In this case, we use 2 QM regions, so that's something that is kind of unique of NAMD and VMD, QwikMD. You select one region here for being the let's say the reading part, like you're reading the anticodon, and then you have the other one is, let's say, the writing, and the writing is where the chemical reaction occurs, and you can calculate even how the force, like how the not force in this case, but like allosteric communication propagates between the two. And you can then use steer molecular dynamics to force the chemical reaction that you're interested in. You can use the string method, also available in MD, like that Emad was saying, to calculate, find the perfect, like the best pathway for that reaction to occur. Then you can use extended ABF to calculate the free energy barrier. And using all of that, you're able to recover the free energy for this system. You're able to show how the reaction occurs. And here is just one nice video of how you see those things. Hopefully that's going to run. Yeah, they're going. So you have here the tRNA, you have the glutamyl tRNA synthetase. So you have an anticodon, you have the reaction center, you have the allosteric pathway, and you're going to zoom in now in the reaction center. You're going to see, yeah, first we're going to show the molecule a little bit. You're seeing how this structure looks like. And then see, these are, we're now going to zoom in there and look at, so these are the parts where these is our QM region, so the region that we're treating the quantum mechanics. All the rest is classical. And you're going to see now how these atoms behave during the simulation. So you're looking at this is the glutamate that is bound to the AMP here, and this is the tRNA, so the last part of the tRNA, the terminal part. And what you're going to look at is are these dashed lines are the reactions that are supposed to happen here. So I'm just representing them with these dashed lines. They're nothing. There's no force there. Just to show what, like, this is for you to look there. You know, like those are the bonds that you're going to change. And what you're seeing here are the orbitals. So these are the highly occupied molecular orbitals. So they're all the way in the back there now. And then you're going to see this hydrogen approaching here. It's going to jump here, and you're going to see all the highest occupied electrons are going to be in this area now. So this area is going to be forcing another chemical reaction, and you're going to see here now, you're going to break this bond and form this other bond. So you're going to bind now the glutamate to the tRNA. So this looks very slow, right? But this whole thing here is happening in, I think, if I remember correctly, 2 picoseconds. So this whole process that you're looking at is just 2 picoseconds. You know? So this is, it's very fast actually. So although QM is very expensive compared to, like, to do one step compared to the classical, at the end, the simulation itself is typically cheaper than the MD simulation, because in the regular MD, the classical MD, we're interested in phenomena that happens at the microsecond time scale, so like nanosecond to microsecond, like we want to go in that range. Well, in this case, we're interested in things that happen in the picosecond to femtosecond time scale. So it's a much faster process. Okay, so yeah, so this is my take-home message to you. Like, you can use QwikMD to assist you in all of that, to do preparation system, do analysis. You can do multiple simulation replicas when it's needed. You know, like, you can use it to study the force propagations that we're showing. You can use to prepare QM, hybrid QMM system. And you can also use to prepare, like, this network analysis to calculate allosteric communications and things like that. Do I have any questions? No.
I have a question.
Oh, yeah, sure.
Oh, yeah, sorry. So I'm totally new to MD analysis, and I'll probably start from QwikMD. And so how can I tell, okay, this is possible with QwikMD, but this is not possible with QwikMD. I have to do MD without QwikMD, but I can kind of graduate from QwikMD and then start working on more detailed MD also. What would be the boundary between the possibility of QwikMD and more like, detailed MD?
For preparing our system, I would say QwikMD can do almost any system. I'm not like to do the, so here, all the way, like in the QM system, for instance, all the way to the SMD, you can do everything with the QwikMD. You're going to the string method, then you're going to use everything that was prepared with QwikMD, but then you're going to have to go into the config file by yourself and fix things and change the way you actually want to do. Because then it gets too complex for QwikMD to have an easy solution for you. Like, so that makes no sense to actually create. But in general, to create your system, the only limitation is I want to do a crazy membrane that QwikMD does not have. Then you might use other tools, you know. But otherwise, for most of the applications, you can use QwikMD all the way through having your system prepared and equilibrated. Maybe then you want to use a method that QwikMD does not give you the config file the way you need, but then it's just changing the config file. It's let's say it's adding a few lines in the config file versus preparing everything. So QwikMD helps you even in those cases. Yeah.
What exactly do you mean by crazy membrane, for example? It's
like I'm just saying, like instead of having a flat POPC membrane, you have something like Emad was just showing a sphere, you know, like that. Okay, we cannot give you that. Like some other tools can give you things like that. Or maybe you have a membrane that is a mix of 10 different lipids. We can prepare that for you, but tools like SharmGUI can do that for you. You know, like so you have to. There are things that you can do outside of QwikMD that might be easier for you, like in a different membrane. But a membrane is just a good example, in my opinion. But like there are probably other things like that that involve modeling steps that you cannot do inside QwikMD, but you can still use QwikMD after you have the model. If you have the membrane this way, I'm saying, you can use that membrane inside QwikMD to add a protein. You can do that. That's not a problem. It's just that it's not going to create a membrane for you. It's not going to model the system for you. It does not have the modeler step connected to software like Rosetta or model, something like that.
I see, got it. Thank you very much.
I have two questions here in the chat. Like one of them is about the analysis that are available in QwikMD. Everything that is available in QwikMD is actually available in VMD. So QwikMD is just creating an easier interface. Okay? So everything that is in QwikMD is actually the VMD tool. Okay? So QwikMD, the idea behind, like I mentioned, is trying to create a hub that is easier for you to use for preparing the simulation and running the simulation, running some simple analysis, like more basic analysis. But like we have the intention to create things like that also for analysis, like having the QwikMD analysis kind of tool, you know, like that all the analyses are in the same place. The other question that I see here is about replicas. You need to rebuild the simulation cell or the same cell with initial velocity. You don't need to rebuild the initial cell like when you're doing replicas. When I'm doing these many replicas for SteerMD, I just restart the velocities every time, like every new SMD. But like even if you don't do that, it works because if you're using Langevin, you have stochasticity in the Langevin term. So after a few steps, your simulation is different from another replica. So like it's not deterministic. How much replica should you do? 3 or more? Okay. That's a tough question. If you're comparing with experiment apples to apples, and if you read some of the papers that we published, like there's the science one that I mentioned here, the results, and there's also JAX in 2019. This one here, if you see my mouse, we were comparing like different numbers, and if you want to see the beautiful plot that agrees perfectly with experiments, you need to do the thing. Your n should be the same as the n of the experiment. I cannot give you a better answer to that. Do you need that to understand the mechanism? No. But if you want to have the same distribution, if you want to recover the distribution from the experiment, you should have about the same n. So if they have dozens, you need dozens. If they have hundreds, you need hundreds. If you want to get the same distribution. To understand the mechanism, you can analyze 2 or 3, it's probably enough. You know, just to make sure you're not looking at a different case. But like in my case, I want to make sure that I'm getting higher force, lower force. If I change one amino acid, we're getting higher force or lower force. And because force depends on the distribution, that is the level distribution, I can go back here. That's the reason why I do so many replicas, you can see. So let's look at this plot here, for instance. Okay, so this is what we call a Bell level distribution, like in a forest. Okay, so I'm pulling. So each one of these is a different color. Here's a different velocity, pulling velocity. Imagine that instead of a different pulling velocity, this was a different system that has a mutation. Okay, that can also happen. It's going to look exactly the same. So if I pull it just once, I might get this guy here. And then if I put just one, the other one, I might get this one. So this is the rupture force. Remember, if you just do once, you might be getting the impression that this is actually much stronger, or even worse. You might get this one and this one. So you might even get the impression that the second one is weaker than the first. So you need enough replicas that you can recover something like this distribution, because then you know for sure that you are reproducing what experiments are doing. And that's where it gets tricky, and that's why I get so many replicas. And to get a distribution like this, like in this case of Bell Evans, I need at least 30 to 40 replicas. So I know it's expensive, but like I'm just pointing out that's how wide we have these things. But then I can make sure that the experiment, they don't even need to do the experiments when we're doing some crazy attacks and different ideas. They only do the ones that we see that are more important. Let's say these are 5 different cases or 6 different cases here, and this is the wild type, and these are all mutations. If I find this yellow one is the strongest one, they can only do the experiment for the last one. All the rest I can do only computation. And because computation is getting cheaper, you know, like with more powerful machines, it's getting actually cheaper to do the simulations and also almost as fast in the experiment. So it's a good balance. No questions?
I have a question regarding the equilibration with QwikMD. You can do that, right?
Yes.
Yeah, okay. So can you adjust the equilibration time manually? And yeah?
Yeah, so like in the
Okay.
easy run, it does not give you any options like that. But in
Right.
the advanced runs, you can do everything you want.
Okay.
Like, it allows you to do everything.
And in the easy run then, what
Easy
is
run, you
is
can
it?
adjust by hand looking at the config file if you want,
Gotcha.
because it generates the config file for you. You can go there and change by hand in the config file. But like through the QwikMD interface, no, because the idea was to create something that was safe, you know, like something that if someone that has no experience with simulations try to use it, they're gonna get a result that is reliable.
Okay, and if it's just, you know, being done automatically, how does the program select the appropriate equilibration time?
Well, I think we just selected, I don't remember exactly how long it is. We just selected one that we saying like, okay, that will work for 99% of the cases, like something like 10 nanoseconds or so.
Okay, so it's like a value, but it's like a value, but not like like dependent on your system kind of situation.
No, it was nothing that complex.
Okay, gotcha. Thank you.
Somebody's asking me here how to introduce constraints in the steer molecular using QwikMD. Yeah, so in QwikMD you can select where you hold, where you pull, and so QwikMD, you tell QwikMD where exactly the atoms that you want to apply force are, and that's gonna prepare the input file for you. Nothing different than running, like doing that by hand. Does that answer your question? Any more questions? Oh, how do I tell an MD3 to QwikMD to use MD3?
You can't. So the answer is you cannot. It doesn't understand MD3 yet.
On your heat, yeah. So yeah, it does not have the flag for GPUs for QwikMD to use like MD3, because MD3 is still like an alpha version. Although we are doing a lot with it already, it's not a finally stable version.
I would even go further, and I would say that really QwikMD doesn't know anything about GPUs. It is written to launch jobs directly that are only CPU type jobs, and even that it uses very simple assumptions about what CPU hardware you're running on. So ultimately, if you're running on GPU accelerated or high-end hardware that's got more than say 8 cores, this is a case where you're better off running it by hand rather than running it through the QwikMD GUI, because that gives you the full flexibility of setting the AMD launch parameters correctly.
Yeah, so basically what John's saying would be my answer for the MD3. You prepare everything with QwikMD. When you get to the end and you hit prepare, you got all the files, you can close the window and not run it through the button, because the button there at the start, remember I mentioned you can copy that to a cluster, supercomputer, whatever you want to use. You go all the way to that point and you copy the files, or in this case, you don't even need to copy, you just launch NAMD the way you want. And you go in the config file and add the line for the GPU mode in case you want to use NAMD3 or whatever you need to adjust. You know, but these are very small steps compared to preparing the whole simulation.
Yeah, this is like the last 0.25% or something. You know, this is
Exactly.
like literally all you're doing is just running it differently, or you're adding, like he says, one line. Other than that, it's basically identical.
Yeah, it's let's say you want to use in a supercomputer center, you're gonna have to create your script to launch in a supercomputer center. There's less work than that.
Or like even in our own lab, even though all of our machines are on the same file system and our clusters can directly write to your home directory, if you want to run a QwikMD job on the cluster, due to the need for it to interact with a queuing system, you still have to run it with a batch submission script anyway. And so even our own people would do it that way by hand for anything that's a real job.
Any more questions? Okay, so I have something else to show. Let me just close up down here. So because my colleague Barry could not join us, I stepped in to help with rendering and showing you a little bit how you can render like this nice kind of images that you saw in my presentation and videos. And I think a lot of you might be interested in knowing how you can actually do those things. And there are many ways, and John is here to tell me all the ways that I'm not doing correctly. The way that you do, there's no incorrect or correct.
Yeah, but I was also here just to give you some guidance too. We had some great questions yesterday, Rafael, and I promised them that you'd blow their minds today. And so it's like simple things like, you know, we don't really explain in the tutorial our typical method for making a water box, maybe, you know, or things like that. There's just nice things that you can probably show really in 30 seconds. It's fun to see somebody actually do it. So I think what you're planning to do is a live demo, right? So
yeah, so like this is my Linux box right now in front of you, and I had two things to show. Like one of them was depending on the time. Like one of them was showing from like how you can show those networks that I was showing, you know, like for force propagation and also allosteric, and using our new network analysis tool. Or in the other part was to show how you can create a nice image, video, whatever you want to do with it, you know, like, and show like the tricks that I typically use. But you were saying that you want to show, but when you're saying the water box, you want me to create a water box like just Solvate, or
just how we like to render them with the glass bubble material. Yeah, you know, so basically just give them your usual, like I think there's a lot of interest in making high quality figures. So, you know, Rafael is famous for going to poster sessions, and people come up to his poster and they say, how did you make that image? And he said, I just made it with VMD. And then they don't believe him. And so this is why you get to see him do it for real. So, you know, he's not fibbing.
Yeah, so yeah, let's try to open one system here, and then we can go from there. So my idea was this system here. This is the respiratory complex that I showed in some of the cases. This is not the most equilibrated step of the simulation. This is one of my initial steps after some equilibration and some minimum equilibration. This is a 2.5 million atom system. You're seeing it look like exploded, but that's because I typically don't wrap my simulations. That's because I like to have the waters this way to calculate if I want to calculate diffusion and things like that. But you can just type a command and wrap if you want. But in my case here, like, so I'm not going to wrap it. So what I'm going to do is to show, just going to show you two things. I'm going to show you the protein. So like you can VMD type protein, and you can see that's my protein. I hit equal here in my keyboard, and you can get closer. And I'm going to create one that's called lipid. So this thing is sitting on a huge lipid membrane. Like I say, this is a 2.5 million atom system, not cheating anything. I'm using a powerful computer, but that's it. You know, like I'm not doing anything special. Okay. And now if I change the lipid to something like QuickSurf, like let's start with something very simple, it's fast. And I change the lines here of the protein to the cartoon. They're gonna look like, please show me something. Yeah, they're gonna look like this. You're gonna see some here we're cutting, kind of like the screen is actually not enough, like deep to show this. So the first thing, we can go there and fix that. And by fixing that, I come to the display settings. I'm gonna change the near clip to 0.01. That's typically how I do it. And Okay, you have my system. I'm just gonna show you one thing first, how you render. You come to File, Render, and then you select one of the multiple versions to render. There are many different things. So if you want to render in the CPU, my favorite is the Tachyon or Osprey version. If you want to run in the GPU, my favorite is the Optics version. And I like the interactive one, so I'm going to click on that one, and I'm going to call it Fast One. Okay, it's going to say it like that, and I'm going to just start rendering. And I didn't change anything, so it's going to be my basic version. And it's going to look like this. Okay. So this is our rendered structure. I'm rendering all the time here live, so it's going to look like this. So many people think that that's what you can do, and that's how you stop. So what can I change to make this look nicer? Okay. So my first thing that I like is I'm not a big fan of the colors of VMD. You know, like they're great for representing things like this fast, but it's not the kind of image, not the colors that I like for final images. So you can come here and you can come to colors and you can come and play with the color. Let's say you want to change the color of the carbon. Instead of being cyan, you can change it to, I don't know, you can change
So
it
I have to interrupt you already. You're using an ancient version of the code. I have a totally revised new color menu. Ah, you're showing the old one.
I know you have that, John, but I'm going to get to my point.
Okay. Yeah.
So you can come here and change that. So,
Okay.
but like the way I typically do, and that's my point, the way I typically do, I don't even use any of that. What I do is I use a script, and that's what I want to show. And my script is just here. So what my script does is I create a lot of color schemes for myself. And like the color schemes, depending on the system I'm working, depend on what I want to do today. Like I want more like pastel kind of colors. I want something very shiny. I want something. So I created a bunch of them, and each one of them is basically this. Like, so this is the neutral one, as the standard one. So this, like every color of VMD is listed here with the RGB code. Then I have another one that's called QwikMD. Then I have one that I call 80s. They're more like shiny colors. And I have one that I call pastel that I use a lot. SDRG, one of the systems, the system that I was showed before that was published in Science. I have one just for that system. So I have one for network analysis. So each one of those are color schemes that I use for the system that I'm working with. And as you can see, it's very simple to get one of these. And the colors that are here, I take it typically from mapping things online. You know, like just use like Google RGB color scheme. You know, like, and you're gonna find like you can find some that are from paintings, famous paintings. Like they're gonna give you the whole palette.
I would also recommend that there's an Adobe app called the Adobe Color app, and it will give you things like a color wheel. And these are very helpful for generating color tables.
And this app also you can point at things and it gives you the color. That's the nice thing about it. The camera, I think it's the same app, right, John? When you point the camera, I have one of those. You point the camera at something. Let's say you go to an art museum and you like the colors. You just point your camera. It gets to the palette out of the image. And so you can get the color palette and replace it here. And then you're going to have the color palette that you want to use in your simulation, in your rendering. So the way I did this then, I created this color palette. It's a one big file that has everything on it. I added that file to my my VMDRC file. Like it's a little bit more tricky, but like you probably at your home directory, you're going to have something called .vmdrc, like if you're using Linux or Mac. And what I did there is just I'm sourcing it every time. I'm sourcing my VMD color scheme every time. Okay, that's my way of doing it. But basically, it is the same as coming here and changing the color, or coming where John wants me to come in the preferences, and then you're gonna have the colors, and you can come here and change colors. You can also come here and change all these colors the same way. So, but then because of the way I did it, I come here and I just type the command that I created. That's called VMD. It's color scheme VMD. There's a lot of VMD, so I started inverted color scheme VMD, and I type, for instance, pastel. And that's going to be all pastel colors. Okay, does not look much difference right now, but okay, I'm changing all the colors. If I render it right now and I call it number 2, you're not going to see much difference other than the colors are different. Okay, and you're going to see there. This is the rendered version. Like I'm zooming in. Okay. So how somebody else uses my command? You have to have my script, but I can share that, no problem.
So everything that you do in the GUI has a corresponding VMD command, and maybe show them the trick real quick. The log
the log
console.
file.
Yeah.
So let's say.
And there's even a GUI for that. If you go in the file menu, there's a log file. Yeah,
I remember. So like I just to point out this here, if you type here log file console, it's gonna show everything you're doing here. Let's say you don't know how to create a script to rotate your molecule. If you type that, when you rotate, see, it's telling you there, rotate X, rotate Y by. So if you type that command, if I now, instead of rotating, if I type rotate Y by 5, okay. Yeah, it rotates Y by 5, see? I can keep doing that command. So like whatever command you want to use from VMD, if you type log file console, it's going to show what the command was here. Okay?
it's a great shortcut if you're feeling like you don't want to read the manual that day.
Yeah.
And it's a good way, like for what Rafael's example, let's say he changes one of the colors in the GUI, it should emit the color change commands too.
And then if you want to stop it,
ask
log file off, right?
Can I ask real quick, the purpose of your color palette is because VMD has an idea what blue is, and you're changing that definition so that every time VMD asks for blue, you get something different. Is that right?
Yes.
Okay.
Yeah,
And the other reason is when he starts doing other types of rendering and shading, you're going to want to change, the original VMD color definitions are great for the type of shading that OpenGL does. It's a compromise for what looks good on different hardware. But when you start doing things like using the ray tracers, as you'll see, it's better to, especially if you use things like ambient occlusion lighting, you get shadows and things. You're going to want to start with much brighter colors so that the shadows don't look overly dark. But I'll let Rafael drive here. So
yeah, so no, that's great, John. Like there are many of these things. Remember, like this is also a lot of the taste. Like this time I'm doing a pastel one. Maybe what I want is something very shiny. Like in all of that you can play. So.
And I would also argue there's no one color scheme that works the best for everybody. And like you saw, Rafael, even for his own work, has several different color schemes. And I would advocate that is probably the right approach. And people end up finding, useful color schemes. Sometimes a particular structure has a prevailing color scheme, so people want to match how that's typically rendered. So for example, Joachim Frank always shows the ribosome with particular colors. So if you're doing a ribosome simulation, you want people to see the same things or be understandable, you adopt the prevailing color scheme other people have been using.
Yeah, also other thing that I would say is important, like if you come here and use color ID, you're going to see the numbers of the colors, and like they refer to a color, like a changing blue, for instance. But also, when you select things like seg name, is going to have an order. And sometimes in that order, I want red first, not blue. So in my head, I completely forget what the name is written here. Colors for me are just numbers. So like I got used to color 0, color 1, color 2, color 3, and I forget completely that my blue is blue. My blue might be red. Like it's just a matter of like what I want to match to that system. Every time I load, it's faster. Like I don't need to worry. You can also map. You can always map things inside, but I find it easier to just change the color. So it's up to you how you actually want to do those things. So
yeah, VMD is not very religious about this, and that's the point is to, and somebody might want to make their image with a whole bunch of different shades of blue. Maybe there's a reason.
The other thing that I like is there are more colors than are listed here, right, John?
Yeah,
like
there's
the numbers
1,000.
here end at 20 something, and like you can get much more than that. I don't remember what's the final number, but I have more than 50 for sure.
Yeah. So if you count the color scale, there's actually around 1,032 or whatever it is,
So yeah, like I use more numbers than just the numbers that are there. Okay, so then if I come here and then, okay, I have my system like this. So the first thing, another thing that I like is I prefer another material. I like my figures. They look more like they're doing with chalky. It looks like I prefer this. AO chalky, so the ambient occlusion chalky material. And I'm gonna do that for both here, and you're gonna just see just changing that. Okay. And then if I just click render again, it's already looking different, right? It's already looking kind of nice, but it's still far from what we want. Okay, so yeah, that's one of the tricks. Let's continue. And like, that's what I do all the time. I render and check, render and check. So now I'm gonna come to that, it's open there, but just to show, I'm gonna go to display session, so display settings, and I'm gonna turn everything here on. So like shadows are on, ambient occlusion are on. I'm gonna turn on the depth of field. And here I can also play with this one, you know, like how much in the back, like how, so like how much I wanna change the back to be kind of white, to disappear, or to appear, you know, like I can play with that. I can also make that linear. Sometimes that's useful, you know, like because the linear is useful if you want to make, let's say, like this. I want to make the protein perfectly visible, but everything that is after the protein starts to disappear. See? And then I'm going to make like this. So it works. It also works. So if I render right now, it's already looking quite nice. And as you can see, some things are on focus now and some things are out of focus, because what I did was I turn on the depth of field. So I turn on this last option here, which is basically like if you have a professional camera and you're trying to focus on the system. So you can focus, you can have how much of the image is on focus, how much is off.
Show them a real-time scrub so
yeah,
they can see.
I'm gonna do that as soon as I
Okay.
change one other thing.
Yeah.
So that, and I'm gonna then just turn this here, the protein that I have new cartoon, which is just color by name. I'm going to change it by seg name. So now I have all the different colors, and I'm going to make this quick surf now van der Waals. Okay, it's going to be a little bit heavier, but it's going to look nicer. So there we go. So now are all spheres. You can change the resolution of those spheres changing here. I'm not going to do that now because it becomes more expensive to render. But like for final product, I always change the resolution.
You know what's funny though? You don't need to, Rafael.
Yeah,
for the ray tracers, they're drawn as a perfect sphere anyway, so it's just eating up.
Good.
So you don't need to do that at all.
But like for the cartoon, I do need, right?
A little bit, maybe up to 15 is as far as I'd go. Yeah.
Especially when you're rendering large systems, and now you see that my image is completely different, right? It's looking much nicer. So I'm in this render mode. I'm in the live rendering. You're rendering while you're talking here. So you're seeing this is looking, so my focal point is actually much closer to me. So if I move now my arrows from my keyboard up and down, so if I go up, I can move my focal point to some part of the protein. You can see that
Zoom is having a hard time keeping up with your screen.
Oh, sorry. Now I move it up and down. So now I move it up, so you can see that now I'm focusing kind of on the protein, right?
Yep.
You can see that the protein is on focus, the rest is kind of a little bit off focus. And if I move to the sides, if I move to the left, I'm actually changing how much of the protein is actually on focus, like how much of my focal point is actually on focus, how much is off. So I can make everything kind of out of focus except the part that I really want to focus. See? I can make just that narrow strip on focus right now.
So what everybody watching should understand is for him, this is happening at about 30 frames a second.
Zoom is not as good as my...
Zoom is only able to draw about 1 frame a second, but Rafael is seeing this almost instantaneously, so it's very easy for him to adjust.
And the other thing that I want to just point out is this machine is not even in the same building that I am. So like you can, all of this is done remotely, you know, like, and you can do that with the Amazon machine, like one of these Amazon cloud machine. But my machine stays in another building where it's like our supercomputer center is. My machine is there and I'm running that from my office where I don't have a hot machine next to me, you know, like increasing the temperature of the office. So, and there you see here, and it's looking nice, right? So it's already looking different, a lot different than before. So people are asking questions here.
So the Blender would be a different beast. Basically, this is a renderer that's built into VMD itself. So this is the optics version of Tachyon that runs on the GPU, and it's doing everything inside of VMD. You can, of course, export a VMD scene to Blender. You're going to be limited to the 3D file formats that Blender knows how to read. But things like the camera parameters, things like that, you would have to type those in Blender because there's not really a file format per se that contains those. And I think somebody else, what was the other question?
About
The other-
what you up and down.
Yeah, oh
and-
yeah, so the
Here.
keys, when you're running the interactive ray tracer, if you press P, it'll print this help screen, or H. P will print the different
I
numbers
can do it
for
right
different
now.
things. Yeah. And then the H will print the other keys, and basically L moves the position of those depth of field. So you can move all
Yeah,
that stuff and
Yeah, you can turn things on and off. For instance, like I can turn off the depth field right now, and you're gonna see that. See, I'm not having the depth anymore. So you can do that while you're rendering. You know, like those are useful things for you to try things. And remember, like you might see these beautiful images and think like, oh, those were like someone that knows how to do it's fast. It is fast to get something like we're just doing now here, but like to get the final versions that we get in paper, sometimes we want to show something specific. It takes a lot of testing, you know, like, oh, I want this better. I want this a little bit different. I want this the other way. And all of that can be done. And now everything that I did here so far, you can also do in a video, okay? And that's relatively easy because you can render the same image in the same way here. So like we select a lot of parameters. If you come here and look, for instance, the distance, the focal point, it's showing for you here, you know, like every time I move up and down. So I move
and if
up
you
and
want to
down.
print, and if you press P, it'll print all the numbers too.
Okay, yeah, so see, like I never do that way, but like John is telling you. So all the parameters are here now. You can see like the camera zoom, how much was the, you can use all those parameters inside VMD to generate the same image without the live rendering, just the normal rendering that's gonna render for you in the background. Now, if you have a trajectory, you can do that for rendering the whole trajectory. And then you have a view out of what you have here. You're gonna have a lot of steps, and then you can use FFmpeg, you can use, I use Photoshop, you know, to generate the final video. Like you can use anything you want to generate the final video.
So when I was telling you guys during my talk that we're going to have real-time ray tracing in the main window, what I'm really talking about is you'll see like what he's doing here, except it'll be that way for everything you do in VMD. So you would be able to have depth of field even turned on while you're just doing other things. You would actually be able to play the trajectory live. So
I'll be able to change the color like right now,
exactly.
Right, which would be awesome. Like I cannot right now. I have to close and open again.
So presently this is all kind of in this special render mode silo, but going forward, it's going to be sort of in the main window. You'll be able to do this all the time.
Yeah. So this is one of the cases. What else we can turn on and off here? Let's see. You might
show them some shiny chrome materials which have reflections.
So like let's now show just the protein, and let's imagine that instead of having this look on the protein, I want to make it shine. You saw the orbitals that I had, like kind of look like a mirror. There are multiple ways you can do it. You can come here and just change to the last one, RT Chrome. But the RT Chrome, in my opinion, is too strong. So what I like to do is to come and create a new material or change the material that I have. You know, like, so for instance, I can come here in the AO Chalky, that's the one that I'm using, and I can create a new one that's called now Material 3023. Material 23.
Because you can please God rename it from something from other than Material 23.
I use it Material 23 all the time, John.
I know. I just want people to know you can, if you have just like Rafael made his list of his favorite colors. Yeah, a lot of people make their own palette of favorite materials, and I would strongly advocate, doing that also.
Yeah, so I do that in some of my scripts. Automatically, I do that in the end to create things for you. And like our network analysis to generate those materials for you, and then when you load, looks beautiful just because all of it was done already. But okay, I change and create a new material here. There are a few things that I like. Some of them that I like is the outline. I really like to see better the protein. So if I render just quickly here, you're going to see it. I forgot to.
You didn't change it yet. Yeah, you got to use it. Yeah.
I can come here and change from AO Chalky to the new material. And then if I click render, you're going to see that let me turn off the. You're going to see that all the borders are now more highlighted. Like they're kind of, it has this black shadow on the corners. I think, in my opinion, that looks nice. So I use that all the time.
Makes it more contrasty when you're printing too. Printing is always difficult So making things look good on paper, sometimes using some of those things is very helpful.
Yeah, so let's say now that for, let's make it more complicated. Let me just get what seg name one of these things have.
So Melanie asked if it would save your new material when you close VMD. If you save the VMD state, it'll save the new material in the saved state. And of course you can copy that out. A VMD saved state is nothing more than a script. So if you have your favorite set of materials in some VMD saved state, you can just copy them out and put them in their own script, and you can just source or play that script anytime you want. So that's an easy way of making your own favorite collection of materials.
So what I'm doing here right now is I'm creating two selections. One of them is just this part of the protein, see? And I'm gonna make this guy different. So I'm gonna use the new material. I'm gonna create a new material, you know, like a new, my material, chrome material. Whatever name I want, chrome material. So like this other one here is going to be my chrome material. And what I'm going to do then, instead of using the chrome, I'm just going to add the mirror effect here. So I'm going to add a little bit and see how it looks. When you do that, if you zoom in there, okay, turn off the depth of field, you zoom in, you're going to clearly see, come on. When you zoom there, you're going to clearly see that this guy now is a big mirror. So everything that is around is reflecting on it. So that looks really nice depending on what you actually want to do. You know, like depending on what, for instance, for the orbitals and things like that looks very nice. If you want something for like a cover image of your paper, that looks pretty cool because you can see the reflection, let's say in the surface, you know, and these are things that I frequently use.
Admittedly, it's purely artistic though.
Oh yeah, it's
So he's not moving the light source in the interactive ray tracer. The directional lights have a fixed position when he's in that interactive ray tracing mode, but he can move them around in the main, like in the main window right now. If he wanted to rotate the lights around, he could. This is one of the things when we get the interactive ray tracing running full time in the main window, you'll be able to change you'll be able to change everything while it's running. Whereas right now you have to run the renderer and then exit before you can change some of these things. So that's the main thing that'll happen when we get that put in, is you'll have the complete freedom to change everything you want all the time.
The other thing that I sometimes use, John, is the headlight that you created for the VR.
Yeah.
I use sometimes without the VR, you know, like because it illuminates the region in a different way.
Yes. Well, it's so the headlight is sort of a positional light that's located at the camera. So it tracks wherever the user's head is. So that's for those of you that want to render VR movies, that's something that's kind of special about a VR movie, is you need to, directional lights don't work as well when you're down inside of a virus capsid. So we have a different kind of positional light for that case.
Yeah. So the other thing that I like is, like when I zoom in there, you start to have that cut clip, you know, like problem, like not really a problem, but like when you're rendering and you can go very close, you know, like you can because now it becomes like this object in this rendering mode, you know, like you can move around the way you want. You can get very close to it, like I'm doing here right now. And back to this is the point where we start the rendering here. I can play with the focal point, like I was showing before. I can play up and down again. I know it's slow for you. I'm just gonna find the point here to zoom, and then I can move here. Let's see. I can have just one helix. See, I can have just one small part that is rendered. All the rest is autofocus. Okay, this is exaggerated, but you might want something. Now, showing this video of the reaction, I was showing that just that bone that I wanted to show wasn't focused. Everything else was out of focus. And that's very useful if you want people to look at one specific point of your video, not everything.
So there are two questions. The real-time ray tracing is going to come in what we're going to call VMD2. So I have it working in a modified version of VMD 194, but it's not done yet. So I'm expecting to make that available, you know, to the public in what we call VMD version 2. And then as far as the focal point goes, you can move it either with the arrow keys or with the mouse using, you know, the F key with the mouse.
Yeah, you can move it up and down with the mouse like this. I'm clicking, okay, to do it.
And yeah, which one is better sort of depends on your, you know, how fast the
I
the
prefer
mouse
the
is.
keyboard.
Yeah, I like the mouse, but I also tend to do, you know, I have pretty fast GPU, so for me it doesn't, you know, it's fast enough. It doesn't matter.
Yeah, I'm using 439s in this rendering, so
Ah,
that's why I can load so fast. The system's so large.
you know what's funny though? You're not actually, so
I'm
even
just
this.
using one actually.
I was gonna say actually the version that you're running can't really exploit a 3090 because it's the old version of optics.
Okay.
So you're running it that when I give you the new code, it'll run about 3 times faster than that.
Yeah, I know. It's already good enough.
Yeah.
Other questions. How do you make that become a video, let's say? Everything that you select here, if you render now, if you come to the normal video rendering,
Like VMD movie, yeah.
yeah, VMD movie maker. If you come here and you select, like you want to render this, the only thing you need to do is like you can generate like directly the impact video. I typically prefer to save the steps like in JPEG or target frames. Like I typically save in JPEGs, and then like you can make it rock, go back and forth. You can make a trajectory. Like, but what I have to do is to come here and change it to tachyon optics, because that's going to be exactly the same look that we're having right now. Okay? Because that's what we are using here to render. Because if I use Osprey, for instance, is another kind of rendering, and I render, you're going to see that it looks different. No, you might prefer, but it looks different.
So Osprey is a CPU based render, and unlike the others, that is written by the Intel guys, and so it doesn't do the materials in quite exactly the same way that the two that I wrote do. And so it's just different, like he says.
Yeah, it looks different. Like you might prefer this one, you know, like for some cases. For instance, there are some of the things that it does. Sometimes I like the Osprey because it looks different, and I like, oh, I want this time to use the way it looks here.
So, for example, Osprey does a totally different scheme for anti-aliasing and for averaging of samples. So, if you zoom in on something, Rafael, so they can see the film grain, let's say.
Yeah, it's not allowing me to zoom in for some reason. What did I do? Well, this K is S, right? So
yeah,
it's not going.
Huh. I don't know. Use the mouse.
It typically works. There might be some glitch right now.
So then use your mouse to scale then.
Yeah, it's not scaling.
Oh, you don't have the mouse wheel?
I can rotate, I can do everything, but scaling.
Weird.
Yeah, I didn't notice that before. So I think it's something right now. But yeah, like you can, depending on what you select here, it's gonna look different. That's just so the tachyon optics is my favorite. You know, like that's the one you're gonna see most of these nice images. That's typically how we do this very nice images. So if you render a video using everything that is set, it's gonna look exactly the same. So if I render a video right now, it's actually gonna look not interactive. It's gonna look like the other one, internal, which is the same. It's just the focal point might be different. So then if I render, it's gonna look a video. It would look like this.
So the difference.
The focal point.
So the difference is the interactive rendering mode is accumulating samples while you're watching it. It's continuing to accumulate stochastic samples. When you run it with the non-interactive, it's running a fixed number of samples per pixel. So the defaults are 12 samples per pixel, and it's shooting 12 samples per ambient occlusion light sample. And so if you're doing stuff that has like depth of field, you might want to crank up those numbers to get higher fidelity. But the defaults that VMD has set, you know, out of the box work very well for most people's scenes and for most movies. And so most of the time you can just sort of leave it as is.
Yeah. So any questions about this? Because I want to show something else just quickly. Good. Out. So like a lot of the analysis that I showed before.
Somebody has a question about wrapping and unwrapping, so maybe you can comment on that when you get into analysis.
How do you affect the analysis, and what type of analysis we don't want to wrap? So it's just like very specific to the kind of simulation that I do. You know, like because I'm pulling these things like very far, and like I want to calculate how water diffuses sometime around, and if I'm affecting that, by not wrapping, I know exactly how like how far each water molecule went, and like it's just easier for me to use that as like the water is just moving in Y direction, as if there was another water box there, another water box there. So for me, it's just easier to use that. Then I can just use VMD to wrap, you know, which is just a PVC wrap all command. You know, like he's gonna wrap everything back to the box because I am loading the box every time. So that's not it does not affect by like by wrapping it back does not affect anything. By having it unwrapped, I can do some analysis more easily. That's why I do it. Can QwikMD append trajectory files with selections? What do you mean?
No. And I know the answer to that because VMD doesn't let you append trajectories presently.
Oh, yeah, that's true.
So we have another tool called CatDCD that will let you do things like that, but VMD at the moment doesn't do that simply because it doesn't write every trajectory format for starters, and some formats are difficult to append to because they have a fixed number of various information in the header, and there's no way to guarantee it'll be self-consistent if you append. So going forward, that's something we're thinking about adding, but it'll only be supported for some trajectory formats. But right now, it doesn't do it for any of them, and for those reasons.
So another thing that I find interesting, like in terms of like rendering nice images, is we last year we published a new tool for doing network analysis. There is one in VMD that you can use. But that one uses, you have to do a lot of the steps, like you follow the tutorial, you have to do a lot of steps in Karma, another software, and it uses Pearson correlation, like to calculate everything. And because we wanted to develop a tool to do much larger systems, a much more complex kind of analysis, we developed a system using generalized correlation calculation, which is a different way of doing. And all of that is much more expensive, it's very different. And we decided to do everything through Python, you know, like because then you could, for many reasons, like we decided to do everything in Jupyter. So everything is done in Jupyter notebooks. You can do all the analysis in Jupyter. You can generate all the data in Jupyter. You can even visualize some of the stuff, like simple things that are not like VMD level analysis, like visualization. You can do that through the Jupyter interface. But then, you know, at the end, you always want to generate the beautiful images out of the pathways and like the nice things about the communities and all this kind of analysis. So what we did is, and that's the thing that I'm telling you because you might decide to do something like that in the future. We created a tool that actually it's a script that generates the DCL script that VMD can read. And when we did that, we created something that every time you run our code to do the network analysis, it spits a lot of files because all these files are generated by the analysis, but also creates this network view true.tcl. And when you load that, what it does is going to load VMD. So now I'm going to show you a simulation with 8.7 million atoms. So this is the spike true, you know, interacting with ACE2. Like it has membrane, has everything here, and it's going to load the network, like connecting the two sides. And I'm going to just show you that, just to quickly show how you can do that relatively fast. And it's looking nice already. So like it's already looking like this. All the colors, all of that was selected inside my Python script. All the colors that are here are matching the colors of the plots that my Python script generates. So like when I'm using Jupyter and I do bar plots for whatever I'm doing there, like the bar plots that are representing this part of the protein are exactly the same color. So all of that you can do, and you can follow our script, is freely available, you can get it. And now I generated this tool here, and that's a nice thing about VMD. You can easily create your own GUI, see, like this. And this GUI is just changing representations and things like that in VMD. So for instance, I want to show the suboptimal path, like it's calculating a path between the two sides of this protein. And the only reason it's taking a little bit long here to load is because this system is huge and I'm loading in different steps. So if I move here, see there are steps of the simulation, you're going to see the pathway moving. Okay? Now I have this pathway here that I'm going to look at. And back to the rendering is, we created it in a way that everything looks nice so that when you click image rendering with the GPU, or just GPU interactive, GPU or normal, when you do the GPU interactive, it's loading everything from VMD, like the way you would click all the buttons, and it looks like this. So the idea was you could generate already your nice looking image without any effort, like without learning much of the stuff. So things like that are things that like we have been talking, like John as well, like John is here, like to make in VMD in the future, like the VMD2, like make some of the steps easier. But this is a way that you can generate your own tool. Like say you want something that always looks in one way, you can generate a script that loads a window. You want to show your collaborator, you want to talk about your system, you can come here and click, oh, I want to show you the step 5 or whatever you want, like a mutation, a different representation. Oh, I want to show transparent. Okay, this is image magic to open this. I cannot change anything. Okay, I want to make it transparent. I want to not make it transparent. I want to hide the protein. I want to show the protein. I want to show nucleic acid. I want to, whatever you want to do. Like you can create buttons that are basically the commands of VMD, and it's going to look like this. And it's very simple to use when you're doing things like your collaborators and to visualize the system at the end. I think you've never seen this one, right, John?
No, I haven't seen it. I was going to say somebody mentioned they had some trouble to run it on Windows, but I don't know if you guys have tried that before. And the other thing I was going to say is I posted the links to the Adobe color tools in the chat window for people that were interested. But yeah, I think, like you say, Rafael, I think that especially when you have a tool that's sort of aimed at solving a particular problem, having sort of built-in bespoke schemes that you've already baked out that work well, it's very nice to have that. And and so that's, we're never gonna
neutral colors, and the paths are always strong colors. It's always gonna look, you're always gonna be able to see it more easily.
Yeah.
Questions regarding the Windows thing? I don't know, honestly. I really don't know how to answer. I need to check. I don't know, did you have problem with the Python part, or did you have problem with loading the TCL? The TCL should work.
That's from Ahmad Adel, or is that?
Yes, that was just thing here, like they're trying to the network view on Windows, but it did not work. Like this TCL
previous, he said previous version, yeah. So that must be the old version he's talking about.
Version, okay.
Yeah.
So
could be something else. Then we'll have to.
Yeah, can be even the karma part. Can be many things. There are many complicated things on that code.
So and Edward Ackett had asked about doing a nice water box. So you can show.
Doing a nice water box. So let me just. Okay, how do I don't have any of the system with a nice water box here. Let me get one.
Well, you can always make one.
I know. Yeah, but I have some.
Show the Solvate plugin doing its magic.
Yeah, we could show QwikMD. That's also nice. Yeah, so
that's true. Yeah, very good.
Actually,
oh, I was gonna mention one other thing. In Rafael's demo, he made a very good point about matching the VMD color scale to when you're doing a plot, like with MATLAB or something else, and you want to use the same colors. I should point out that both for MATLAB and Python Matplotlib, we have scripts for VMD that will make the VMD color scales exactly match those that are used in these other tools. And so that way, if you're rendering a figure and you want to color the structure with exactly the same color ramp that you used in a plot, that's something that's very easy to do. And in the newest versions of VMD, newer than what Rafael is showing, the default list of color scales now includes all the major ones that are found both in MATLAB as well as Matplotlib. So the ones that are considered high quality, that are good for visually impaired people, colorblind people, all that stuff, those are now built in as default schemes, like the Viridis color scheme, for example, Plasma, various other ones that are very popular. So, go ahead, Rafael.
I just generated a system with QwikMD. As you say, like John was talking, I was able to do everything. So this is just a basic simulation, and you have this box that is showing like this, which is not how you want to see it, right? But because it's loaded, you see 40,000 atoms there. I can just come here in the regular VMD and I can just, 1 second. I need to get rid of the box, which I can do by, provide the command, John. What's the command to get rid of all the drawings?
Draw, delete all.
Draw, delete all, that's the.
That's something that QwikMD drew, I gather.
Yeah, it is. I
Yeah.
was trying to remember what was draw. I remember it was something related to draw. So then I can come here and do the presentation. I have the new cartoon one there, and then I want to make one that's called water, and then it's going to select all the water marks. This new cartoon, you're going to see anything, and then you can see show lines. You're going to see that's my water box. So then you want to see my ice cube. What I do typically, I come to quick surf. There are multiple ways you can do it, but I come to quick surf. I increase the radius. I increase the radius to get more flat, as you can see. You know, like it doesn't get so much details. You can play with these numbers here, like as a surface. You can play with grid spacing. All of that is going to affect how exactly it's going to look, you know, like and how flat it's going to look. Then I come to.
I usually like having a smaller grid spacing, so it's a finer mesh. It's not as bumpy.
Sure.
And then if you want it, but if you use the resolution, you know, the radius scaling is what'll make it, you know, smoother overall. You know, that's a good way of making it smooth.
Sure.
And then, so you can decide how bumpy it looks. And you might ask, well, why do you care, right? And the answer is when he chooses this glass bubble material, for example, you'll see that how bumpy it is has a big effect on how much of its silhouette you see when it gets rendered.
So now if I come to the display settings, I'm just going to turn on the things here. Like before I was doing with my scripts, so like I'm going to go come here, turn this on. And then I have multiple ways of doing it. I can do transparent. Now I'm just going to do transparent first. You can see how it looks. And if I come to render and I use the Packing Interactive, and that's going to look like this. So this is my water sphere now. You can have this glass bubble like this. Then it's already looking quite nice, right? But like you can change it. You can change the color of the blue. That's like a tone of the blue. You want something light, like lighter tone of blue. That's something that I typically do. Let's say you come here, find the best blue. That's typically how I use. Then I can make the color of the protein also
Complementary.
Yeah, to get more contrast. But then what I like is I can make the protein glossy, and I can make the water. Okay, first of all, let's create a material. That's how I typically do it. Materials. I come to Talkie. I'm going to create a new one.
Yeah, there's nothing religious about any of the default colors or materials in VMD. They're just meant to be a starting point. You know?
Yeah, actually, you can go there and change the original one. It's just I'm not doing that for you to see.
Yeah.
So what I do then, I take the AO Chalky that I like, I reduce the opacity, which is going to make it transparent. So if I come here and I change this one to material 23, which is a new one, let me just turn on. It looks a little bit better.
Yeah.
So you see, it's like this. So I can change how much I want this transparent, how little I want this transparent. I also like the outline and the outline tools like this because it's gonna show the ice cube better. And then when I render it, rendering button here now, a lot of things. When I render it, it's gonna look like this. So then you can have the ice cube the way you want. That's what you were looking for, John, or?
No, so that's a starting point. Now do it with the glass bubble material instead of this one. So I think this is great. This is the best you can probably do with a normal transparent material. But we added a special glass bubble material which uses this angle modulated transparency, so that when you look at it face on, like if you orient it, it's mostly transparent, tremendously transparent. Even if you make the edges very visible, now you can see the interior very easily. And in fact, if you make this more bumpy, that's another interesting one. This one is very interesting with a bumpier water box. So if you make the resolution of the thing, finer, there you go, like that. So that's a great example. Now render it.
And you can make the disappear the one inside.
Yeah.
Use water. It's gonna.
So now with that glass bubble material, you see, now you can see the interior very clearly. And as you rotate this thing around, no matter which direction you look at it, when you're looking straight on, you get a mostly transparent surface. The only things you see are basically the silhouette of the various bumpy parts and so on. And of course, you can still use your depth of field tricks and everything else to do all kinds of other stuff. So I think those are the two predominant ways of doing water boxes, either using a conventional transparent material that's just like, you know, like a film, you know, or balloon or whatever. And the other is that glass bubble material.
Yeah, I typically prefer this one because I typically do that for larger things and I want to show it like the whole structure. And I think that one makes the transparent. I don't know. It's a taste thing.
Yeah.
This one I know it looks like almost like if the protein was outside of the glass. You know, like
yeah, it's just depends on what you're trying to show. You have to do, these are things that the point is there's not one answer that you have to decide as a scientist what it is you're trying to communicate. And you know, the benefit of this material, I think, is like, especially when you're making a movie and things are rotating around.
Make sure it's gonna look nice always.
Yeah, it sort of has a predictable behavior no matter which way you look at it. That's all. So yeah.
Any other questions?
Yeah, so now you guys should ask questions. How do you do some other trick? If you guys have seen some of Rafael's gorgeous images, now's your chance to ask him how he did that.
Yeah, so the other thing that I like to use is the view change render.
Yeah, you just went over it. It's up one. There you go.
So let's say you want to make a movie where you want to rotate around. Like, so other thing is, a lot of these more complex movies are actually many steps of what I'm going to show to you right now, you know, because you combine tricks. But let's say you want to make a movie here that save viewpoint. You make a movie like you save this viewpoint here, and then I'm gonna zoom in here like this, you know, like, and then I'm gonna save this viewpoint, and then I'm gonna rotate whatever like this, and then save the viewpoint, and then you wanna make the glass bubble go off, and then you're gonna save the viewpoint, and then you're gonna zoom in, I don't know, rotate again, save the viewpoint. So I save all of them, and I'm gonna add all of them to a movie. And like, I'm just using the standard here, 0.5 seconds per step. So like, I'm going to enable video. Now, if I go to the movie maker, I can use it there, but like, I can show it later. If I preview here, see, this is going to preview like what my movie is going to show. See, like, my movie is going to now show. I go there, come back. It's drawing the box again because it's interfering with the QwikMD thing, but like, forget about it. So if you do that, so this is your video, like steps of your video. You can come here and change how much time you're going to take between position 1 and 2, 2 and 3, 3 and 4, you know, like 4 or 5. This is the number that is next to it. Then you can come here in the visualization, take the movie maker, and instead of using like rock and roll, trajectory, you can use definer procedure. And the user defined procedure is what you define it here. So you're going to use whatever you define it in this step. That's enabled now here for Movie Maker. It's gonna be your steps here. So when I render this video now, it's gonna render exactly what was created here, and then you can generate these different angles and different views, and then you start combining things. And like, you make maybe one scene that you go all the way to one point, and then you make another one that you change focus, because this cannot change focus for you, but you can easily write a script that change the focus. You know, like, and then you start from there.
I was going to say that one of my suggestions, you know, Rafael already mentioned that he likes to emit the individual JPEGs. And if you're going to make a complicated movie, I would definitely advocate to do that, because if that allows you to then import the complete movie sequence you just made out of VMD into something like a Adobe Premiere or one of the Apple tools for editing movies, you know, Final Cut Pro or one of the others. And once you've done that, you know, then you can use the video editing tools to add labels and, you know, titles and all this other stuff. And then you can make very nice, high quality movies. And that's a very easy way to work. You know, the built-in video compression stuff is very machine dependent. Like if you have the wrong version of Linux, it may have an out-of-date version of FFmpeg. So in general, I tend to favor doing things using real video editing tools and just saving all the raw frames. You can't go wrong by doing that. That's a good idea.
The other thing is, for instance, the video that I was showing that I changed the focal point, that I did everything with VMD. You know, like I was changing the focal point with VMD. But if you have one JPEG with one focal point and another JPEG and another one, there are tools in these programs that take you from one to another. Like they just merge, like they create a merge between the two. And then you create a whole trajectory of going out of focus to the next one, and you don't need to render again, you know, like, which is faster. So there are many tricks like that if you start using this other software. So it's nice to have all the frames. It's also nice to go there and add labels, to add these final things. You can go, in the future, you want to use that slightly different for some presentation. You can have it. You can have multiple videos because you want to have in your presentation, pause and talk about it, click again. You know, like all of that can be done if you have all the JPEGs.
I also, yeah, and I like to start by, you know, if you're if you have enough compute horsepower, it's nice to make the transition times in the VCR plugin relatively long. So instead of real fast transitions like you did there, I'd make them longer. And then in a video editor after the fact, you can always speed things up, but you can never slow them down. Or if you slow them down, you start to see it clicking, you might say, or, you know, you run out of frames to do that with. So it's better to render more frames than you're going to need, and then let the video editing software, you know, play those back faster, or to cut sooner or later and things like that.
Yeah, well, John suggesting you come here and change the time to a much longer time. That's if So like you could do that because you're saving like 24 frames per second. I think that's what it is.
That's
so
the default. Yeah.
Yeah. So if you do like running 24 frames per second, you're rendering 50 times 24 images here. If you want to do like 100 now seconds, even if you want a shorter movie later, it's easier. You know, like you can do that. If you're going to stretch it, it's going to look bumpy. It's going to look not so nice.
And, you know, we did the entire chromatophore movie using view change render and some scripts. And, you know, we did render a lot of the clips on a supercomputer rather than a desktop, but the concept is the same. And, you know, using the scripts you do here, you can render them in parallel. Or you can just let a single machine and GPU crank along rendering the scenes. You can render them in OpenGL rapidly, you know, using the interactive display, or you can render it with the ray tracers. You have a lot of different options, so you can do rapid previews, different things like this. Yeah.
We talked 40 minutes more than what we should. So any questions?
No, that was great, Rafael. I think it was a really nice demo. I have some more things to show maybe in the afternoon. I don't want to keep people waiting for the lunch break or whatnot. So yeah, let's do that. Let's stop now and do like a 45 minutes break.
Do you wanna tell them for the Windows users, we posted an updated version, and those of you running Windows who want to run the FFTK are going to need